Now we are going to study about biosynthesis of flavonoids. We have seen the biogenesis of lignans in the previous class, and lignin was also formed with the help of para-cumaric acid, which was obtained by cyclic acid pathway from phenylalanine. So similarly, the flavonoid synthesis also starts in the similar way. If you see, this is the cyclic acid pathway in which this phenylalanine is getting converted into trans-cinnamic acid, and the enzyme used is 1. So here I have written the enzyme 1, that is phenylalanine ammonia oxidase. So phenylalanine ammonylase will convert phenylalanine to trans-cinnamic acid. So what is basically happening here, one ammonia is being released, ammonia molecule is being released, and it is converted into the trans-cinnamic acid. This trans-cinnamic acid is getting converted to para-cumaric acid, that is 1-hydroxy group is coming on the para position, and the enzyme which catalyzes this step is cinnamate 4-hydroxylase. So cinnamate 4-hydroxylase catalyzes the conversion of trans-cinnamic acid to para-cumaric acid. Now this para-cumaric acid, in the presence of 4-coumarate coenzyme A, ligase gets converted to para-coumaroyl coenzyme A. This step we have seen in the biogenesis of lignans also. So this para-coumaroyl coenzyme A, along with 3 molecules of mannonyl coenzyme A, and in the presence of enzyme chalcone synthase, gives us chalcone. Chalcone have this kind of structure. This we have already discussed in our previous class. Now this chalcone is one of the flavonoid, and this chalcone only provide us different flavonoids. Like if this chalcone undergoes changes in the presence of chalcone isomerase, isomerization will occur and the ring closure will take place and flavonone will form. So if the isomerization occur and ring closure occurs, then you obtain flavonone in the presence of enzyme chalcone isomerase. Now this flavonone, in the presence of flavonone 3-beta hydroxylase, flavonone 3-beta hydroxylase के presence में flavonone will get converted to dihydroflavonol. That means a hydroxy group will occur, dihydroflavonol. So 2 hydroxy groups will be present on the phenyl rings. I have not assigned the positions because various different positions are possible and it's a subclass. So all the possible dihydroxyflavanols will come in this category, which can further be converted into flavonols, this kind of structure. Now if you go on to the right hand side, chalcone can be converted into deoxychalcone. That deoxychalcone can further be converted into isoflavones. So isoflavone will have this kind of structure. In this case, the B ring present on the second carbon shifts to the third carbon. So this is isoflavones. Now flavonone. This flavonone can give us flavones as well as flavin 4-ols. Okay, so if this carbonyl groups get converted into alcoholic group, then we get flavin 4-ols, and if unsaturation comes here, then we get flavones. Now if you go to the dihydroxyflavanols, you can get flavin 3,4-diols. This carbonyl group will get converted into hydroxy. That's a possibility. So you can get flavin 3,4 diols. From this you can get flavin 3-ols. One hydroxy group is being reduced, then you get flavin 3-ols. And if you condense this molecule, condensed form, we get tannins. So that depends, the structure of tannin will depend from this what the starting material is. In this case you have many hydroxy groups attached here, but that will particularly depend upon which flavanol you are taking. Okay, now from flavin 3,4 diol you can also obtain the charged molecule, that is anthocyanidins. In this case you get a aromatic ring C, in which oxygen will have a positive charge, and if this further can be converted into anthocyanins. So this is basically the complete biosynthesis of flavonoids. It also starts from the cyclic acid pathway similar to the lignans, and on formation of chalcones we have various possibilities. Different flavonoids can be obtained depending upon the various reaction steps followed. So this is biogenesis of flavonoid.